mostly doing at this time, which involves mainly study of water distribution systems, and a gang of people are involved in this. The goal of this project is fairly straightforward. The goal is to understand the makeup of drinking water distribution system microbiology. You knock down a glass of water, you're knocking down in the order of 100,000 bacteria per milliliter. And that's a good slug of bacteria. If I tell the students, you have to think of it as a little extra protein. The problem, of course, in doing this is the high degree of variability that one has to expect. There's a long history of people studying microbiology of water systems using a variety of methods. And although using culture-independent methods, we're certainly getting a much closer view than we've ever seen. But the issues of variability are great. Different water sources, feeding distribution systems, different disinfection regimes of the different systems, the meta properties of the systems, and age of the system, water handling, et cetera, et cetera, et cetera. So there's a lot of variability, and what that means is you need a lot of different samples. And so the goal is, at least in principle, simple. You sequence a lot of DNAs from drinking water distribution systems, and you have to look at a lot of systems with very different properties to understand things. And sequencing has to involve more than simply high-volume sequencing. We've used 454 and MySeq currently, and Sanger has not gone out of style, as I will point to you. We need longer sequences. So the sample processing is pretty straightforward in this case. We're going to look at a lot of different sites around the country, and how do you do that? Well, you don't contact the water authorities everywhere because you're never going to get anywhere. They don't want to hear about this stuff in any case. So we mostly use what we call guerrilla sampling. And you go into a community and you pick a couple of publicly accessible places, and you go into a restroom, for example, and you run the water for a little while until you measure the chlorine residuum. And then when the chlorine residuum flattens out, or if the temperature flattens out, then you sample a few liters and you're out of there. Then you filter down the DNA and do sequencing in a variety of ways. Now I note in passing that what we sample is not the ecosystem that we would like to sample. What we sample in this way is planktonic material. Some of this will be planktonic organisms that like to be planktonic, but most of it probably is stuff that's being knocked loose from biofilm scattered throughout the many, many, many miles of any particular system in the locale at which you have to be sampling. There's a lot of line pressure flux, for example, in all of these systems, and so this is going to wash stuff off the walls. And you have cars driving around 6 feet above, and that's going to create local micro-earthquakes, which is going to shake things off of the walls as well. So what we sample is not what, in principle, we're interested in. We sample planktonic, we're interested in biofilms. Well, all we knew when we started this was that there's microbiology in there. The concentrations are going to be 10 to the 7 to 10 to the 8 cells per liter, something like that. You know that there are no coliforms or other fecal indicators because that's what the EPA says we have to know. Beyond that, you knew nothing whatsoever about the water that you're consuming, wallowing in continuously. Now here's what you see if you take a sample of this and stain it with a DNA binding dye, DAPI, and look at fluorescence. You see lots of stuff in there, and you can't see it very well from this perspective. But if you suck it down onto put it onto an electron micrograph, put it out on an electron micrograph, you can see that not only do you have a lot of different cell types, but also a lot of other sleaze that's in there. Presumably the stuff that as well washes out the biofilms. We mostly don't know much about that at all. So we've sampled quite a few settings around the United States now. We've studied Colorado systems in good detail. Jim Ross, graduate student in the lab, used to be a graduate student in the lab, but studied multiple samples, many samples from 2 Colorado systems using different disinfection regimes over a 2-year longitudinal period. From that, we found out that every time you take a sample and analyze the microbiology, it's going to be different. Every time, every sample, even in the same system, it's going to be different. But if you do a lot of samples, generalities begin to emerge, and you have to do that. So a bunch of sites we've looked at, I'll tell you about a couple of things. The big issue is that you have to have a lot of samples in order to get some idea of what general microbiology is. So I'll tell you about one of these studies, one of the geographical surveys that we've done along the Arkansas River Valley and the lower Mississippi rivers, and we'll be going into another system soon to study other aspects. Now, you'd like to learn something about what a general good system is. So Eric Hollinger and Kim Ross fixed up a little recreational vehicle to be a small laboratory. And they started in Salida, Colorado, and drove off down the valley of the Arkansas to the Mississippi and then south to to New Orleans, taking samples along every few hundred miles along the way for a total of 17 samples. 2 samples were taken at each site, in each city that was sampled. They reached New Orleans, turned around, came back, and again sampled the same tanks. So we're getting 4 samples in essence for every particular community sampled. Now, we selected that path for a couple of reasons. One reason is that all of the river drainage is in river sediment position. We don't have a lot of different geology there, and so we don't have a lot of different geochemistry coming into the systems. So we would consider these to be very clean systems, but we are also interested in the potential health-related aspects. As I will tell you that the biggest problem that I see in terms of microbiology of the built environment are mycobacteria associated with water distribution systems. And that's a pretty big deal. You might not have thought much about non-tuberculous mycobacteria, but non-tuberculous mycobacterial pulmonary disease is a big deal, and it's getting bigger. It's increased about 3-fold over the past 20 years. The reason for that, there's even very little data on incidence because it's hard to diagnose and the docs aren't looking for it. Some data have accumulated from Medicare results, and here's a recent paper which gives you a shading map for the frequency of distribution of diagnosed NTM pulmonary disease, probably at least an order of magnitude be up below what is really there. Now, and in some areas you can see that in the southern United States it's up over 200 cases per 100,000. In some areas it's over 300 cases per 100,000, and that's reaching epidemic proportions. And so it's a pretty serious issue, and this is almost certainly coming in from the water distribution systems. Who's there? Well, you go out and you take a lot of samples, and the first thing you want to know is, well, did I take enough samples? As I say, these are all very complex communities, and just take enough. And so now you play various statistical games with that. And so these are summations of the 4 different samples from each of the sample sites. And as you see, each of them in the collection, we're getting 50 to 100 taxa, something like that. More on what taxon means momentarily. And if you look at the level of the saturation, if you will, of the system, that so with goods collectors curve, you know, we're over 95% saturation with respect to most of the communities analyzed, and overall in the distribution system, we're doing pretty well, something like 400 or so taxa, 50 to 100 in any particular system. So I think we're getting a good idea of this system network, these systems. So now, word on bacterial taxonomy. Bacterial taxonomy is really screwed at this time because of the simplicity of the classic taxonomy that's used. So, for those of you who don't think about that, we have the 3 big domains, bacteria, archaea, eukarya, and within the bacteria we have, for example, about 100 of these phyla, only about 30 of which have any cultured representation whatsoever. So the database is very sparse that's out there. Now below that class, order, family, genus, species. And when you collect a whole bunch of sequences, you drop them into some kind of computer program that will look at a collection of sequences and parse out what your particular sequence is closest to and give you a call. You go into environmental genomes, environmental DNA sequences with typical next-gen sequences where you have maybe 300 nucleotides, something like that. Most of your calls are going to be in the order of the family area. That's almost no specificity whatsoever. Some, but not nearly enough, because here's what you want, and that's what you don't get. Moreover, in the microbial world, the species even is a very, very big place. If you look at a collection of organisms you consider to be the same species, they will share only about 30% of their genes. Another 30% will be distributed widely within that, quote, species, and another 30% will be seen elsewhere in biology. So there's a very big disconnect between our understanding of microbiology from the laboratory study into the field. Very big difference. Now, if you just look at the large level, the phylum level, for the microbiology that we see in water systems, it's quite simple. So one's going to see only about 5 of the main bacterial phyla, and within those even it's fairly simple. And more detail on that. So if we now begin to look at details, so these are just a series of taxon calls, 17 sites, and then the frequency. As you can see, that each of the samples itself is fairly simple, but between the collection they're very different. So lots of mycos, no mycos. What's going on here? What's the variation? You can see idiosyncrasies of systems. For example, here's Thiotrix in a system, and you can predict that that one has a lot of sulfide in the water. It happens to be Baton Rouge. You'll note that I didn't identify any of the samples that we were taking. They're all de-identified. So then the identification issue becomes a significant one, potentially a legal issue that we all need to think about. So it's a relatively simple collectively. Main organisms for any given system will be fairly simple. And as you move from sample to sample, even in the same system, there's quite a bit of variability. So for example, this is the Morrissey-Warren so-called plot, which gives sample-to-sample comparison, both within a particular city being sampled, so a lot of these guys are all alike, and you get over this sample set here, you see relatively little similarity, even among the different samples in the same city. These are just examples. Sort of represents what you do see. Sometimes it's reproducible more or less within the system, other times not. The important point is you need a lot of samples. You can also do other things with these kinds of taxon calls. You can play games like, for example, two-part analyses where you can look at biases in one type of community versus another. For example, surface versus well water. There's a lot of significant variation. This is just another way of presenting the same data, where you have chlorine versus chloramine. Yeah, you see differences. High versus low nitrate, yeah, you can see a few differences there. And these are all a lot of other metadata that fold into them as well. Most of it we can't make a whole lot of sense from yet. Now, here's the problem. We're getting extremely low-grade information. In these next-gen sequences, we get typically around 300 base pairs, something like that, and that just doesn't take you very far. You're never going to get species-level calls. We can't do phylogeny with the sequences because they're so short. Moreover, sadly, these various pipelines in dealing with what we call pyrobabble tosses out anything that doesn't fit the training set. That means that everything that you might be interested in is different is out the door, and you never get to see it again. And I don't care that it's getting better, but you better archive your DNAs for the next gen technology, because you're probably going to want it. And I know a lot of DNAs aren't being archived, which should be. So it matters. You can't stop at 300, if it matters, you can't stop at 300 base pairs or probably even ribosomal RNA. So in mycobacteria, the big issue is organisms, Microbacterium avium, by sequence, approximately ribosomal sequence, approximately indistinguishable is Microbacterium cordomi. That's going to potentially have problems with pulmonary disease. This is not known to have such problems. Or folks are interested in learning about Legionella pneumophila, Legionnaire's disease, Pontiac fever, and that sort of thing, versus other Legionella. You're never going to see that by ribosomal RNA, in the least because we don't have any database of what the environmental examples of these kinds of organisms are. And the environmental database is at best sparse. So the important point is that we need to be very careful with taxonomic realities. And I carved a little bit about that earlier, about invoking names that might, you think, invoke interest in the public. It doesn't invoke interest, it invokes fear. People think they're drinking pure, unbacterial contaminated water. At any rate, microbial diversity is a much, much, much bigger place in its complexity than our nomenclature will admit, and certainly our pipelines will admit. But the public does care, and we need to be careful about that.
A while ago, Lars and I worked in your lab with you on MTMs in the hospital therapy pool setting. 37 degrees and they said absolutely low. What is the relationship between what you're finding in this temperate water system and those that we would find in like therapy pool, hot tubs? Because those really grow.
It's also so the question is, we've previously studied mycobacteria occurring in a variety of public places. For example, as Scott brings up a private therapeutic swimming pool where they had a number of cases of identified Microbacterium avium pulmonary disease. Now, the technology we were using then is different than this. This is next-gen sequencing, 4x4 sequencing in the case of this set. And previously, as you know, we were doing Sanger sequencing, had a lot longer sequences to deal with. But also, in retrospect, I don't think we had very good taxonomic calls because of the lousy database, Scott. So I really can't make a real comparison at this stage.
Norm, maybe you covered this, but you said you took two samples. They took two samples on their way down and two on the way back. Were all four taken in exactly the same place?
They were taking two different places in each community.
So each pair was 2 places.
Each pair was two places.
So when you showed that figure with the
More
diversity,
seat of horn,
it
yeah.
wasn't sorted in a way that we could see where they were in the same place. It was just the same city or.
Well, it was actually buried in that figure. We can talk about this a little bit more.
But in those comparisons, was there more similarity in the same place? And less?
Depends. And some of them, yes, and others are dramatically different.
I mean, I think your whole point was that one of the things that really matters is place, and
That's right, and the volatility of it, which is one important thing of that comparison as well.
I didn't see it. Did you see a difference between the chlorine versus chloramine disinfection?
Yes, both qualitatively and quantitatively. Specifically with chloramination, you see significantly higher diversity, typically about twofold higher, something like that, in different suites of organisms.
And with respect to the microbacteria, did you see a difference between the two?
The general observation is the more oxidative you get, i.e. the higher the chlorine concentration, the greater the microbacterial
There are
concentration.
survivors.
I think there's the
Yeah.
real selection. These drinking water distribution systems are really extreme environments. They're dark, they're cool, they're enclosed, they're wet, of course, and loaded with poison, namely chlorine or chloramine. Chloramine has a longer persistence in the distribution system, which the water folks like, because it keeps the water clean longer. And however, Kim has also studied the microbiology that goes out of the processing plant, and it's very different than what shows up at the tap. So it's the ecosystem itself is in the piping system itself. And we don't know a whole lot about the variation within it. We don't know anything about the variation within a distribution system, although it's probably a lot of what we're seeing.
So presumably, you sort of hinted at this, either longer reads or metagenomic data theoretically could provide the finer scale resolution. Do you have a feeling for whether or not there's enough reference information to use to do metagenomic analyses to compare to, or are we still left with?
Yeah, I don't believe there is, Jonathan. There are some things. Now, the hooker is you're digging into a community that you know nothing whatsoever about. And so with ribosomal RNAs, we can be pretty confident that we're going to get pretty good selection of what's there. But then when you start going with other genes based on cultured organisms, you're not going to be in very good shape because you can't predict reliably PCR primers. Even where you can, we've invested quite a bit of effort in trying to use the internal transcribed spacer between the 16 and 23S genes to pick up a better definition of what are these various microbacteria. So you can PCR from a myco 16S forward primer and its 23S reverse primer and pick off the ICS. But from all the sequence information we've seen from the environmental stuff, we can't match it to anything because there's no database. It's a real problem.
So taking back to building science from gene sequencing. So Jordan talked about damp houses. And a house could be damp from evaporation of water that comes in the potable water supply. It could be damp from exhaled breath and other things that people bring in. It could be damp from water, from groundwater that's getting up, or from rainwater. So there's three very different sources, or at least three, of moisture that can result in a damp house. Do you have any thoughts about how that might matter?
Only the gut reaction which Jordan talked about, namely more moisture, more bugs.
But no distinction based on the type of water, the source of the water.
I think we can't say anything about that.
Is it worth looking at?
Probably. One thing we've done a study of household humidifiers.
Yeah, oh yeah.
And there you really begin to see the difference between using distilled water on one hand as a source of fluid and tap water on the other hand. And then those humidifiers are real serious.
And cleaning those ones.
Yeah, yeah.
Thank you.